Welcome back. We're gonna carry on with a lecture from Saad Jbabdi, which unfortunately could not join us today, but he provided us with a recording of his presentation. I know also about structural parcellation of the brain, so I hope I'll be able to answer a question about his work. But we'll see how this goes. Thank you very much.
Hi there, my name is Saad Jbabdi, and this is a OHBM educational session on brain parcellation with structural connectivity. Let's start with making a general point about understanding systems, and this in particular in neuroscience. In order to understand a system, we need to break it into its constituent components and to study their interactions. And the brain can be understood at multiple levels: molecular, cellular, circuits, and regions. And at each level, it helps to be able to identify the components and their interactions. So at the finer scale, the components are molecules, then at a cellular scale, there's cells and circuits. But in terms of systems neuroscience, one question that remains to be answered is, what are the components units? What are these brain regions? And the first thing one might think of while looking at the brain is that its appearance is quite striking. It has cortical folds. And one question is, are those folds relevant for subdivision of the brains into its component subunits? And the answer is not quite. First, there is large amount of variability between people. So while some of the major folds are consistent between people, the minor folds are very inconsistent between people, and in fact inconsistent with functional areas. And so we can't use cortical folding to identify borders between brain regions. And this was noted quite a while ago by, for example, Alfred Campbell, one of the fathers of neuroanatomy. I'm going to read the quote: "Save these primary fissures, there is really no practical naked eye guide to the localization of the different areas. For all the so-called secondary fissures bear a very inconsistent relation to given fields and cannot be utilized as fixed boundary lines." So over a century ago, it was already known that cortical folds don't map with function very well. And these great anatomists instead turned into myelo and cytoarchitecture. Here I'm showing a cartoon depiction of three types of cortical areas: primary sensory cortex, association cortex, and primary motor cortex. As we know, cortex is subdivided into different layers, but the relative size and importance of these layers differs between functional regions. For example, in primary sensory cortex, layer 4 is much more prominent than in motor cortex. And so people have used cytoarchitecture as a guide for defining functional borders. These are three examples. As you can see, between different operators, you might end up with different types of subdivisions depending on what features of myelo and cytoarchitecture are driving the studies. This is an example of Brodmann areas 44 and 45 here in the lateral prefrontal cortex. The Jülich lab, and in particular Catherine Amunds, have developed automated and kind of user-independent ways of defining borders between areas using, for example, silver stains, stains for cell bodies, and looking at cortical profiles across the cortex in terms of this intensity stain. One can automatically detect changes in the stains between areas and find borders. in this way. This is a validation that these borders indeed are consistent with functional borders. These are brains from different individuals, and the green area it depicts anatomical V1 as predicted by a cytoarchitectonic atlas. In fact, one of those atlases derived from cytoarchitecture in the Jülich lab. And the red-yellow borders are borders between V1 and V2 as determined using retinotopic studies in fMRI. And so, as you can see, the green area is surrounded by the functional borders. So, what we know is that microstructure correlates with functional borders, but gross anatomy and cortical folds don't. However, these histological studies can only be carried ex vivo, and if we want to be able to define cortical areas in living humans, we need a non-invasive technique. So how do we do that? Well, let's go back to the question of identifying component subunits. So it was framed in this way. First, identify the units, and then study how they interact and how they are connected. But actually, one can frame it both ways. Perhaps by studying the connections, this will help us identify the component subunits. And that's where the idea of a connectional fingerprint came from. So this was a term proposed by Dick Bassingham back in 2003. And what I'm showing here is a figure from that paper, looking at connections of macaque prefrontal areas. And in particular, if you zoom in on area F3 and F5, you can look at the connectivity profile to other areas in the same part of the brain. And the point here is that these fingerprints are very different. And so perhaps by estimating connections, we can then use that information to drive the differentiation between brain regions. Okay, so last time I gave this talk, it was in person in front of a class. And so I thought I would add some quizzes to make it a little bit more fun. I will do that here, but nobody will answer my quiz, so I'll answer it myself. And this is just to hammer the point home. So the quiz was, if on the left-hand side, I'm giving you the connectivity profile of a brain region to all of these other regions in the macaque brain, can you identify the areas with this particular connectional fingerprint? And the answer is area V1, because we know that it has very strong connections to adjacent area V2 and V4, and weaker connections to other areas. And so, with the premise that we can use connections to identify brain regions, now the next question is, how can we measure connections? Well, before MRI tractography, there are only 3 ways in which we could measure connections. 2 of those are available in humans, lesion studies and dissection, both post-mortem. And in animals, we can also do tracers, and in fact, this is the gold standard technique. And then in the late 90s and early 2000s, tractography emerged, and it kind of fits in this box here. It has, I would argue, the same properties in terms of accuracy and spatial resolution as dissection. What's tractography in a nutshell, in case you didn't go to one of the other educational sessions in OHBM on diffusion? Tractography uses the fact that diffusion of water is anisotropic in the white matter, so it's more hindered across the fibers than along them. And by making those measurements and inferring, we can infer the orientation of axons in each voxel and reconstruct their trajectories throughout the brain. What does tractography offer? Well, it's an in vivo technique. It's non-invasive, and we can use it to study the whole brain at once. And so unlike maybe some other techniques like tracers, where we can only study one connection at a time in a given animal, here we can study all the connections at once in a brain. And because it's in vivo, we can compare these connections with other measurements of function and behavior. And so we can use tractography to answer other new questions that maybe other more invasive techniques can't answer. But tractography also has issues. It's low resolution, so the voxels are much, much larger, 3 orders of magnitudes larger than the axons. It's indirect. We are measuring the diffusion process, not the axonal trajectories directly, and therefore it is error prone, both because the MRI is noisy, but also because our models are perhaps not perfect representations of reality. And it's also difficult to interpret quantitatively in terms of, say, the number of axons that connect A to B. But nevertheless, it also has all these pluses that one must not forget. Now, with tractography, we can look at something I call the connectional contrast. And one of the earliest examples of such contrast is the study of the thalamus. So if you look at the thalamus in a T1 weighted image, it has very little contrast between the different voxels. But if you run a tractography experiment from the voxels in the thalamus, for example, here from a voxel in potential ventrolateral nucleus of the thalamus, you get the green tracts here connecting to M1. And if instead you see tractography from an adjacent voxel, you might get a very, very different tractography answer. And so in terms of tractography, there is high contrast between these two voxels, but little contrast in terms of T1 weighted signal. And so using this contrast, you can identify borders between regions in the brain. And there are 2 categories of methods to parcellate gray matter using connectivity. The first category is hypothesis driven. So for example, we know that we are looking for connections to regions A and B, but these connections differ in the so we have connections between A and B that differ in the connections to C and D, etc. So we know exactly what connections we are looking for that are differentiating A and B. But if we don't have such hypotheses, so for example, our knowledge of anatomy in humans is much poorer than our knowledge of anatomy in animals, then we can let the data speak, and that's called a hypothesis-free or data-driven clustering, and we'll see examples of that as well. Now, coming back to the thalamus, we know that nearly all incoming sensory information is routed through the thalamus. We also know that some of the cortical connections or communication goes through the thalamus. And we also know that different nuclei relay different kinds of information. And so we'll have very different connectivity profiles. And this was used before to parcellate the thalamus into regions that connect to, for example, S1 or M1 in cortex, or premotor cortex, prefrontal cortex, temporal cortex, etc. Here's a more specific application of such parcellation of the thalamus, and it concerns this nucleus here called VIM, or VIM. And one way to define VIM using connectivity is as the intersection of these 2 tracts, one descending to the contralateral dentate nucleus in the cerebellum, and the other connection going up to the ipsilateral hand motor area. And in this intersection of these 2 tracts, likely located in the VIM. And Harith Akram used this strategy to define VIM in patients, in Parkinson's disease patients. And it so happens that doing deep brain stimulation in this part of the thalamus is very effective at reducing tremor in these patients, as you can see here. So this is the DBS target. This is the patient before the stimulation trying to write, and this is the patient after the stimulation. In terms of hypothesis free, so looking at all the connections without a priori target regions that we're looking at, this is illustrated here. So this is a brain, and I'm looking at the connections of the blue region here to the rest of the brain, and the red region here to the rest of the brain. And by looking at all the intermediate locations between the blue area and red area, one might look at transition in these connectivity profiles. So these connections look more like each other. These look more like each other. And this part of the brain has a transition which corresponds to this border here between the two regions. So that's the kind of hypothesis free. Let's look at all the connections and see how they change rather than the connections of specific to specific targets. So here's one of the earliest examples of such application. And it concerns looking at the medial prefrontal cortex, so this part of the brain, and looking at the border between SMA, so the supplementary motor area, and the pre-SMA. And it was kind of known before that this happened to coincide more or less with y = 0 in the MNI space. So the goal here is to define a border not based on y = 0, but based on the connectivity profile. So as I said, this is hypothesis free. And so what you can do is construct a matrix where the rows of that matrix are all the voxels in an area that comprises both SMA and pre-SMA. And the columns of that matrix are all the voxels of the brain. So the values here in any given row are the connectivity profiles of a given voxel in this red area here. And then what you can do is cluster this matrix. As one way to do it is to calculate a cross-correlation matrix. So looking at the correlation between any seed voxel and any other seed voxel. And this matrix can then be clustered, or the rows and columns can be rearranged to find structure in the matrix. And here, if you do that, you find these two prominent clusters, which can then be projected back onto the brain, and that gives you the border exactly in the right place. So the blue voxels here have a connectivity profile that is very different to the red voxels. Same technique has been applied to many different cortical areas in subsequent years. So now that we have this way of identifying borders between regions or clusters in connectivity matrices, one may ask the question of how many such clusters. So if I have a brain area and I want to subdivide it into multiple brain areas, how many clusters can I find in the data? How can I find the number of clusters in the data? Well, the number of clusters will depend on many factors. It may depend on the tractography method that is used. So for example, deterministic versus probabilistic tractography, or global versus local tractography, etc. It may depend on the definition of connectivity profile, and we'll come back to that later. It may depend on the similarity measure. So we need here to inject a measure of similarity between connectivity profiles, and the results may depend on the definition of that similarity measure. It may also depend on the clustering algorithm that is used, and whether there are borders to begin with. There's no simple answer to all of these questions, and so one needs to validate the number of clusters that one gets, for example, by looking at reproducibility or reliability of the results, and also testing against other sources of data that aren't tractography data. So here's an example, a study from 2009, looking at parcellations of the cingulate cortex. And in this study, they found that 9 clusters that you can see here had the highest reproducibility between subject reproducibility. They also used meta-analysis of task activation profiles for different types of tasks to validate the location of their clusters. Here's a study that looked at the insular cortex and looking at the connectivity profiles throughout the insular cortex. And the question was, again, how many clusters? But what this study found is is that the insular cortex, unlike, for example, SMA and pre-SMA, the insular cortex had smooth variations with no clear borders. And one way to find that out is to use low dimensionality representation of these connectivity profiles. So in this case, using something called spectral clustering or spectral reordering. And the basic idea is to take all the voxels in the insular cortex and calculate their connectivity profiles, which is which is the connectivity to the rest of the brain. So this is a hypothesis-free method. And then take all of that high-dimensional information into a low-dimensional 2D space where the distances between points represents the similarity in the connections. And you can see a very smooth, almost one-dimensional transition between the front and the back of the insula. And if you contrast that with the SMA, pre-SMA, you find two very clear clusters. Okay, so this idea of parcellation doesn't just apply to the gray matter. One can also do parcellation of the white matter, and one can also do it in exactly the same way. So there's a hypothesis-driven way of doing white matter parcellation. For example, I want a tract that must pass through region B, but not region D, etc. Or you can do it in a hypothesis-free, data-driven way. So an example of hypothesis-driven is to draw, as I said, regions of interest and say, if I want the green tract, I'm going to see it from the whole brain and only keep the streamlines that pass through A and B. And this is the kind of thing that was used early on in tractography to identify major white matter bundles. Here is an example with the arcuate fasciculus from Michael Catania's work. A data-driven technique could go in the following way. First, we construct a bunch of streamlines, then represent their trajectories in some sort of low-dimensional space in which you can identify clusters. Apply these clustering back to the tracts to get your tract parcellation. And this is work from Lauren O'Donnell. Here's another kind of more recent way of doing this. The idea here is to take a connectivity matrix profile. So for example, from gray matter to white matter. This matrix can be decomposed into the product of two matrices, here using independent component analysis or ICA, but other types of matrix decomposition can be used. And what this gives is not only clusters in the white matter, but also clusters in the gray matter that are associated with these white matter tracts. So for example, here you can see the corticospinal tracts associated with areas of the sensory motor cortex. And here's the arcuate fasciculus. Okay, so now that we've seen ways of doing gray matter and white matter parcellation, I thought I would do an aside, fun application of gray and white matter parcellation. And this is a comparative anatomy application where we're trying to look at whether we can find ways to map areas in the macaque brain on the left to areas in the human brain on the right. So the premise here is to use, or the idea is to use connectivity to do so. So if you look at the white matter in a lot of the primate species, in particular monkeys and great apes, a lot of the tracts, the major white matter tracts are common between these species, but the cortical projections can be very different. So for example, here, the arcuate fasciculus in the human has a large number of projections into the temporal lobe. These get smaller in the chimpanzees and absent, so, at least for the lower temporal lobe in the macaque. So can we use this to do this work of comparative anatomy and finding homologies in the cortex? This is another one of those quizzes. I've forgotten about this, but let's do it. This quiz hammers home the point that some of these tracts can be identified that are identified in the human can also be identified in the macaque. So you've got a fronto-occipital pathway on the left-hand side, and your job here is to find the pathway in the macaque that best matches. No surprise, it's A. So and that is true for a number of different pathways can be identified in both species. We've written a tool called Extract, which is in FSL, which allows you to reconstruct a number of different pathways automatically in the human and in the macaques with matched names. Once you do that, what you can then do is produce a connectivity profile for each and every voxel in the cortex. So for example, here, the areas highlighted in yellow have strong connectivity to the arcuate fasciculus, whereas the areas here have strong connectivities to the middle, longitudinal fasciculus, etc. So each cortical area has a tract connectivity profile. Here are some examples of these tract connectivity profiles. So on the x-axis are a number of different white matter tracts, and the blue and the black lines correspond to the profiles of these dots in the frontal cortex. So once you do that, you can then this provides a common space for comparing macaques and humans. So you have a matrix in the human where tracts are these extract white matter tracts, and you have rows are cortex. So the tract space is matched across the species, but the cortex isn't. So for every part of the brain, you can then use some sort of similarity measure in this common tract space to find a match in macaque. Here's an example of that principle in action. This is human area MT, which can be identified using a myelin sensitive measure throughout the cortex. The tract connectivity profile of this human area MT is shown here in blue. And then if you look at the macaque connectivity profiles, you find the red area here, or the red voxels here, are the voxels that best match this particular connectivity profile. And that corresponds to the predictions for where this area MT is based on histology work. Okay, so we've talked about parcellation of white matter and gray matter. We've talked about some applications in, for example, DBS and in comparative anatomy, so that's great. Now let's look at some caveats. So remember this slide where I showed the pluses and the minuses of tractography. So let's remember the minuses. Tractography is low resolution, it's indirect, error prone, etc. And so we have to be very careful when using it in applications. And one way to be careful is to validate our results, for example, against known atlases, against other types of contrasts. Maybe we have some hypotheses from functional studies or some data from cytoarchitecture to validate these parcellations. So here are some examples of all of these four things. This is the thalamus again, and the red and blue dots represent centers or peak activity profiles from a bunch of different studies from a meta-analysis of executive tasks and motor tasks. So what you can do then is look at where these dots fit in with the parcellation of the thalamus from connectivity. And that's what's shown here. So the red dots all fit in nicely in the putative mediodorsal nucleus, whereas the blue dots, so the motor tasks, fit in nicely with the sensory motor parts of the thalamus. Here is a validation of the SMA and pre-SMA parcellation using fMRI task activations. So here the red blobs represents activity to finger tapping task, whereas the blue dots represent a activation to a serial subtraction task. And the border, the functional border, is exactly the same place as the connectivity border, which gives us some confidence on the results of the connectivity analysis. And finally, here's a validation using cytoarchitectonic atlases. So here are the results of the parcellation using connectivity with two different algorithms for doing the clustering. And this is the Jülich atlas, the cytoarchitectonic borders. One point that I wanted to come back to is this idea that the results of the parcellation depends on the features that you give it, the connectivity profile that you give it. And to illustrate this, I'm showing two results here for the same part of the brain, the sensory motor strip. On the left-hand side is what parcellation you would obtain if you were to use all the connections to the rest of the brain. And here the subdivision or the parcellation is dominated by a dorsal ventral axis. However, if you restrict your connectivity profile to the thalamus, and so just looking at the connections between this part of the brain and the thalamus, then the parcellation is dominated by a anterior-posterior subdivision. And of course, both are correct. The anterior-posterior subdivision corresponds to sensory versus motor subdivision, whereas here it's more a primary versus secondary cortex. And so both are correct, and therefore one has to be careful when doing parcellation that the features that go into the parcellation, the connectivity, is going to drive the answer of what parcellation is. But one doesn't have to see that as a problem, but rather as a feature of this kind of work. And so remember this example with the VIM nucleus. We injected hypotheses there by saying the VIM is defined through the connections to the hand area and the dentate nucleus. And so, of course, the injection of these hypotheses drives the parcellation, but this is a good thing rather than a bad thing. Also wanted to come back to this point that there aren't always parcels. Sometimes brain areas are continuum of connectivity rather than clusters of connectivity. And this is most striking in sensory cortices, which have maps of the outside world inside the cortex. So for example, the somatosensory cortex has a continuous map of the body. The visual cortex has continuous maps of the retina, and the primary auditory cortex has a continuous map of sound, frequency of sounds. And these continuous maps are likely to be correlated with a continuous connectivity map. So remember the case of the insula where we couldn't find a border throughout the insula. And so it's kind of an open question what to do in those cases, how to best model the cortex, whether we should cluster or not. So yes, so there are open questions. Of course, there are technical open questions. The imaging techniques we're using, so connectivity through tractography, are low resolution and indirect. And there are some fundamental limitations. For example, how to reconcile different techniques. What if functional connectivity and structural connectivity give us two different answers? And what to do when we don't have sharp transitions between regions. And I'll conclude by again citing the great Alfred Campbell. I'll give him the last word. And so I'll just read out the quote: "The process leading to the accomplishment of functional localization in the cerebral cortex is such a complicated one and involves so many side issues that perfection cannot be attained or even hoped for until the fruits of investigation in a number of departments are thoroughly weighed, sifted, and assorted." Or in a nutshell, parcellation of the brain is hard. That's all. Thank you very much.
I think we won't really have time for questions. And anyway, Michel will also talk about white matter and has quite some expertise into tractography and diffusion. So I suggest that we gather all questions for both talks, Saad talk, and Michel talks at the end of Michel's talk. So yeah, that's my pleasure now to introduce Michel. We're gonna talk about the parcellation of white matter based on functional MRI. Yeah, so that's gonna be, I'm sure, a really great presentation because Michel has a very long expertise in the field. Looking forward to your presentation.
Thank you very much. So now we've seen how to divide the white matter anatomically and divide the cortex based on the structural connectivity. And in a recent work, we published these maps where we divide the white matter based on task-related functional activation that happen in the cortex that we decode with the white matter. And for this educational, I thought I'll walk you through how to do this kind of decoding so that you can run it yourself on your own time if you like. So this is white matter, but most of us are more familiar with this kind of representation of the brain that are just simple axial slices of the brain where you can easily spot the difference between the gray matter and the white matter, the gray matter being gray and the white matter being white. But the white matter is not like this simple structure that is of blunt organization. It is actually way more complex than that. It is made of wires that connect at very high speed, around 300 to 350 kilometers per hour, different areas in the brain. I often do this metaphor when I'm in the train between Paris to Bordeaux, or any kind of high-speed train. You reach at those kind of speed, and this is probably how an action potential feel when you like onto an axon. You go at that speed between different brain areas. In this slide, so you look at the structure of white matters have been cut into slabs. If I get a mic, I can walk you through it. You can. Hello? Okay. You can see that the white matter is organized in different structures. You have main connections that you see like here in red and change their color because here it's coded according to the orientation of the white matter. So structure here in the middle are connecting the left hemisphere to the right hemisphere, and very essential for the integration of the functioning between the hemispheres. But you see also like some of those blue structure over here that are cut, and those are the projection system, and they're mostly corresponding to subcortical nuclei projecting on the surface of the brain, as well as a spinal cord that project to the surface of the brain, and also the cerebellar projections. And you have also those structures that are in green, that are high-speed connections between regions within the same hemisphere, and they make like quick associations between regions that are far apart in the brain. And when I started studying white matter, I was always wondering, what do you need to draw wires that long between brain regions and not put them next to each other if that's not because there is something critical happening that requires the association, the fast association between those brain areas. In a nutshell, what I'm trying to tell you is that connections in the brain are critical for the functioning of the brain, even way more than just conducting the signal from A to B. Actually, function might eventually emerge just from this interaction rather than just a conduction. If you're interested into connections and roles and meanings and method and how to study it, I've been reading a few books about it, and I thought like, because this is an educational, and we're always looking for good reading for the summer, I'll walk you through why I like those books, and so you know, if you're interested in one domain into studying structural connectivity, which books you should buy and read this summer. I'll start with my book with Marco Catani that I obviously liked because, you know, I wrote it. But this is a book that will tell you about the anatomy of white matter connection and the history of white matter connections, as well as divide those connections into different systems and their functional role based on what we knew before 2010. Then there is this pair of books from Olaf Spons. In the network of the brain, you'll have a depiction of all the methods that can be used to study connection in the brain macroscopically, as well as a very nice introduction about the history of studying connections. In his other book, which is Discovering the Human Connectome, you have an explanation how to use graph theory and what graph theory really means in the studying of connections. We heard a little bit about graph theory this morning, but it is an interesting way to study connection because it can capture the structure of the connectivity and help you identify modules in the brain and association in a way that is different than just looking at the anatomy of the brain. In sum, if you look at the graph theory, you get rid of the neuroanatomy, and instead of having things that are far apart but highly connected in the brain, you'll bring them closer when they are technically highly connected. So you get rid of anatomy and you look at it from a different perspective, which is connections and eventually timing, because connection is decreasing the timing of synchronization. I read this book, which is called Cerebral Cortex. That might be misleading because, like, when we're talking about white matter, it's called Cerebral Cortex. But it is actually a wonderful book that brings together architecture, connections, and a model of brain evolution, which is called the dual origin concept. It is really probably the most advanced anatomical descriptions of connections in the brain and main rules of the relationship between the cytoarchitecture of the brain, the way the cell are organized within the 6th layer of the cortex, and as a connection between brain regions. And it also gives you a nice model to think about to do comparative anatomy between rodents, non-human primates, and humans. You have the fiber pathway of the brain. It's also a wonderful book. It's dedicated to the axonal tracing in the macaque brain. So you'll have all the details of 20 to 50 years of axonal tracing in the macaque brain condensed together into this book with wonderful text describing the anatomy and the behavior of macaque. The book of Sebastian Seung, I Am My Connectome, How the Brain's Wiring Makes Us Who We Are, is a more conceptual book. He's been doing a lot of electron microscopy, and his goal in life was to reconstruct a mice connectome, which he didn't achieve because now he moved on to the private industry. But the book is interesting in the concept that is behind it, which is you not really your cortical activations, you really the way your brain is connected, and the way you experience life or you learn new things will be a different way you weight those connections between neurons. Fundamental of the Brain Networks Analysis, it is really a book that will explain to you step by step how to do graph theory with support of software. So if you want to throw yourself into graph theory, read the book, do it step by step. At the end of the book, you're a professional, you know how to do graph theory. That's basically, they did the job. It's a great book. And then you have The Entangled Brain from Luis Pessoa that is coming out next November 2022. That is also a really good book where he's talking about this entanglement in the brain where function might not emerge from the single activity of a brain area, but through the interaction between brain areas, and function being the emergence of this interaction. So if you read all this, you can come here and give my talk. That's what I'm saying. Now, All right, so this is how to study connections, or why connections are so important. Well, you don't have to believe me, but you certainly should believe these people. So David Van Essen in 1982, if I remember well, 1983, sorry, said that area can be arranged in a well-defined hierarchy on the basis of the pattern of interconnections. It really means that the connections are driving the organization of the brain. As a matter of fact, he drawn those beautiful circuits here where regions are arranged based on their connections. If you're more a Marcel Mesulam person, you will read that he said that nothing defines the function of a neuron more than its connections with other neurons. And well, you cannot be more extreme than this. Essentially, he's saying that really the way you are connected are gonna define what you're gonna do. And then if you're more a Carl Ziller's person, maybe a little wiser, you read that he said afferent an intrinsic connection as well as cellular type. And their properties are the structural basis of a brain region's function. In sum, we spend so much time studying the cortex, why it really looks like the solution is in the connections. And mostly we've been doing that because for a very long time we didn't have any way to study those connections until in humans, in the living human brain, until we had diffusion-weighted imaging. Yet, nowadays, all our atlases, even the best one published in Nature, such as the one of Maglassa in 2016, we will have the presentation this afternoon, give a great precision and sharp information about the division of the cortex, that they will flatten, but nothing about the white matter division in terms of function or functional territories. So we thought, how about we try to solve this? And we brought our best tools to try to solve this. We thought, like, if we want to divide the white matter Bayesian function, we need the largest data set that give us information about how function distribute in the cortex. So we took neurosynth. We went onto the website, hacked the website, downloaded all the maps. Then we thought like we can get to use the best tractographies that we can have to get the anatomy of how things are connected together. So for that, we downloaded the Human Connectome project at 7 Tesla from the diffusion-weighted imaging from the Human Connectome project. It's 180 subjects. It's free of access. You can have it. And we process it with our best tool, which is Star Track. So we spend decades adjusting our code to have a tractographies that match post-mortem dissections. I don't want to fight with anybody about what is the best tractography, but our belief is it should match post-mortem dissections, and that's just where I'm going to leave it. And there are like other people who prefer to use tractography to extract as much information as you can from it, from a computational perspective. But this is not what we do, and this is not the school where we're coming from. We're anatomists, so that's what we use. We created matrices of how things are connected using the parcellation of Maglassa, which is a very interesting one and sometimes underestimated. So if you open the supplementary material of this paper, it's about 150 pages of analysis and demonstration and supplementary material. Each parcel is described and linked up with the literature. So we spend a lot of time on that. It's really worth reading the supplementary material of this paper. All this is freely available, so you can do it at home or over the summer if you like. And all the links are also available in the program of OHBM, as well as for all the presentation this morning. All right, how do we do that? So we thought what about if we were seeding randomly the brain thousands of times in all the subjects to produce for each seed a probability map of connections? Simple. You can do that. We have a software that does it for you. We usually use it for brain lesions, but here, let's imagine we have random seeds. Or should we? Because if we think about it, where are those functions coming from? You know, how did we define function? How did we think about episodic memory being different from semantic memory? I think it's fundamentally coming from the fact that we met patients that had a brain lesion and had a problem, and we were like, whoa, what is going on? And then we thought maybe he has a problem remembering about his past, episodic memory, or he specifically has a problem into remembering what an object is, and there'll be more semantic memory. So there might be a bias in the way we understand function, which is linked to the distribution of lesion in the brain. And so we added this level of questioning, which is, are we going to have a better decoding of function using random seeds or using lesions that you have in the most frequent time, which is stroke? If you take stroke, the distribution of stroke is highly biased. There are areas that are more often damaged than others and must have bias the way we define functions. And in order to control well, we created a random distribution of lesions. The code is also available with the paper, of regions that are exactly the same size as a lesion, randomly distributed, but respecting the distribution between left and right hemisphere as well. So it is matched in everywhere except that it is randomly distributed, and that's where you see that there is no red here. It's because it is randomly distributed. And for each of those regions, we convert them into map of connections, probability of connections, or disconnections in the case of lesions. So we then further characterize as a matrix using the Mad-Glasser parcellation plus subcortical areas extracted from the AAL3 atlas. Because if there is something missing in that parcellation, it's subcortical structures. And subcortical structures are really important, so don't forget to use subcortical structures. The lesions were provided by Parashkev Nachev. That's why his picture is on this slide. It was just an acknowledgement. So thank you, Parashkev. All right. And then those matrices of deconnection, that's funny. Why is it upside down? There's a conversion between Mac and PC. Anyway, don't pay attention to that, not yet. So you take the components of your matrix of connections for all your seeds, and you put it in a principal component analysis with a varimax rotation, and you try to see how much variance you can explain with that. If you take the random seed, with one component, you explain not that much variance. With two components, you explain a little more. And you carry on like this until you reach a new cap about 85% of the variance of your connectivity being explained by the principal component analysis with about 30 components. I think to reach 100, you need to reach 100%, you need like 70 or 80 components. With stroke lesion pattern of connections, if you run your principal component analysis, you'll see that you always explain more variance. It's because they're not randomly distributed, so it's easier to simplify. And you reach a cap of 99.5% of the variance being explained with 43 components. And that's great, because you really simplified your problem from an infinite number of dimensions to 43, which is great news. And then what we did, we were like, okay, those components are profile or disconnection, ingredients of disconnections that repeat themselves. We're going to see whether we can correlate that with pattern of activations in the brain, meta-analytic activation from neurosynth. So I do apologize if that's upside down. You have the activation pattern over here, and you have the log score of the structural connectivity in the x-axis. And you have this systematic correlation between each of the component and a specific cognitive domain of activation. And so I was like, wow, this is cool. Is it better or worse if I use random seeds? Remember the question, are we biased by lesions? And so we compared, and you can see that actually, when you use lesion in the brain, you will decode better functions than if you use random seeds. And the main reason is the way we conceive function has been highly biased by the distribution of lesions. And this little difference that we have here is this bias that exists. So we took in our advantage to build our atlas of function of the white matter, but ultimately, we should fill that gap. So those were numbers, but you know, kind of like to see some brain. So what we did a regression of the loadings of each components onto each of the map of connections to create statistical map of the connections that were related to each of the components. And we did it twice to assess replicability of the findings. So when you do that, the map that you have a correlation of 0.8, which is solid replicability. So you remember I showed you, like, you know, the slide with the text that was upside down. So I showed you correlation with functions. Those are the same example of functions and components that I used here. So the component number 1 correlated very well with activation in the involved articulatory functions. And when we look at the white matter that was related to this component number 1, is mostly the arcuate fasciculus plus U-shaped fibers connecting the insula. No surprise here in the left hemisphere. This is a track that we talk a lot about with language. In healthy controls, changing with language, but also in patient when it is disconnected, leading to aphasias. The component number 6 was related a lot to navigation. And what we had essentially are connections between the V2 area and the retrosplenial cortex and the hippocampus cortex and the hippocampus, of course. This is a system, as you know, in the hippocampus. You have the spatiotopic maps. Connecting it with the visual cortex is probably a good idea in order to orient yourself and navigate in space. So again, here we thought like it was a good candidate of connection supporting through this integration the function. For activation related to the eye field, so we didn't find any connection with the parietal eye field, but we find this strong interhemispheric connection between the left and the right front eye field. I want to stress that the level of significance of those maps are thresholded extremely high. So we like around t-value of 10 over here and 30 over there. So like there are also extra connection contributing to that. I'm just showing you the top contributing connections. But the association between the right and the left front eye field is essential for the coordination of the gaze. And as a matter of fact, if you have a patient that show a stroke damaging the front eye field, it's not only one eye that's going to deviate, those are the two eyes. And that shows the level of interaction that you have between these regions. Another example is component 12. for calculation. And you'll be like, this is funny because these look really very much into the gray matter of the intraparietal sulcus. That's because our eyes are biased to the gray matter. This is truly in the white matter. Those are all the U-shaped fibers that connect the intraparietal sulcus, the top, and the inferior part of the intraparietal sulcus. And in terms of acalculia, you got this paper of Andreas Kleinschmidt, who describes that the Gerstmann syndrome that include acalculia is associated to the deconnection of the U-shaped fibers of the intraparietal sulcus. So in a nutshell, those components are correlated with functions, and when you look at their anatomy in terms of connections, they seem to have a link as well with symptoms, or what we know about what those connections should do and what kind of symptom you should see when they are deconnected. But this is only 46 components that we talked about, and I show you only 4, but there are way more functions than that. And we thought maybe those components, like the example of the soup that I took this morning, are just the ingredient of main functions that combine together to give you those extra functions that need the interaction between these components. So we flipped a little bit our matrix of analysis, and we looked at, for each of the task-related meta-analytic activation, the profile of correlation with the components that we use as a regressor to produce map for each of the neurosynth cortical map inside the white matter. That we did twice to check again if that was replicable or we just modeling noise. And here we had a correlation of 0.88 between the two data sets. And this is how we ended up in this, because we had 502, if I remember well, map of white matter related to specific terms. So I'll spare you time and will not show you those 502 maps. We summarize here into this top contribution map that you see here, but each individual map is downloadable from NeuroVault. And you find like this contribution, for example, of the Perisylvian white matter to language through pseudo-word, phonological fluency, and the contribution of the same Perisylvian white matter in the right hemisphere being more related to impulsivity. In the core of the brain, where you're going to have the phonics over there, you have maintaining and binding information together through association. You can really have fun and get lost into these maps. But we were like, this is the top of the iceberg. It might not be really fair to just give to people an atlas, which is one dimension, and where we show that there is only one function per track, which is absolutely not true. You have many functions involving those tracks. First, we assess the effect size of those prediction inside the white matter that we use as an error, which is like the goodness of fit between our model and the prediction. And here you can see like the entire brain is of high effect size, but except for some structures that are like here, like in a portion of the corpus callosum, and a little bit in the cerebellum of there. You have also the pons that was not very well predicted. And then we put this cap at a middle effect size, 0.5, and we try to see how many terms are loading on each white matter streamlines to give us what we call versatility maps, which are like how many functions can be associated to the white matter for each of the streamlines. And you can see here that you have a strong asymmetry in favor to the left hemisphere, mostly the language system, where you see that you have so many more terms and functions that have been studied and accumulated for the left hemisphere of the arcuate fasciculus compared compared to the right hemisphere. Here is the top part of the arcade fasciculus, you can get up to 70 terms, while in the right hemisphere, you're happy if you have 3 or 4. And that's mainly because we know so much more about language than we know about visuospatial processing, impulsivity, and emotion. We have such a sharp division and granularity of the different level of language that we have, and we have no equivalent for the right hemisphere. And that require also more work. That's it. You have here the list of all the data and results that you can download for free, all the softwares and the code that allow you to calculate the effect size maps, as well as the code that allow you to create random parcellations that are matched with any parcellation that you want in terms of size of parcels and distribution. I want to thank these people that have always been super helpful working with me. And I just want to remind you that we have this special issue in brain structure and function that Sarah and I are gonna handle all year, where you can submit any new parcellation, any new idea of parcellation, or update on a current parcellation that you just saw that was published a few years ago. Maybe it needs a little update. We'll be happy to receive that as well. Thank you for your attention.
Yeah, thank you very much, Michel, for the very educational presentation. I think we can feel the professor inside you. That's great. And this very long experience of reading a lot of books. I really like this view of really going deep into one topic by reading a lot and going into the literature. Okay, we have time for questions. So yes, there is one over there.